So my talk today, I was actually asked to give this talk to the family session. And so I'm going to talk to you about all these potential new treatments to treat homocysteine related disease, but I need to make it accessible. And so I'm assuming you guys are what it says, which is a family with homocystinuria. And I'm going to try and keep it very simple, but I have to give you a lot of basics, okay? So the talk organization is shown here. I'm going to spend the first third of the talk giving you an introduction to the genetics and biology of inborn areas of metabolism. And once I do that, you'll have the tools then to understand when I talk about what is chaperone therapy and how it works. What is enzyme replacement therapy? How does that work? Gene therapy, and the hot new one is called gene editing. So we got to go back a ways. We got to go back to this guy, Gregor Mendel. He's the father of genetics, and if you want to know the truth, he's my personal idol. He lived in the Czech Republic, and he bred pea plants. And he discovered in the breeding of these pea plants some weird stuff going on. If he took these purple pea plants and he bred them to each other, he got this weird situation where a quarter of the pea plants were white and 3/4 were purple. And he did this with like 8 different traits, and he always got the same ratio, this 4-to-1 ratio. And he came up with this idea that there are these mythical, he didn't call them genes, he called them factors, and that every time you bred something, it was getting a factor from its mom and a factor from its dad, and everything had 2 factors. And he said, well, the ones that are white, they're getting these little b factors that are recessive, that weren't expressed if the big B was around. And he published this paper, and in 1860, nobody knew what he was talking about. It was ignored for the next 40 years. The next big discovery was around the turn of the century. Some guys in Columbia University, this guy Thomas Hunt Morgan, was studying fruit flies. So we went from peas to fruit flies, and no humans yet. And what he discovered was that fruit flies had the same genetic inheritance patterns that Mendel had previously shown in peas, but he also was able to realize that these factors were being carried on their chromosomes. Fruit flies had big enough chromosomes that you could sometimes see where there were changes, and then they could correlate the changes with the changes in eye color in the mice. He won the Nobel Prize in 1933. This work was probably done around 1910 or so. There's always about a 20-year delay between when the discovery was made and when these guys win the Nobel Prize. Mendel never won a Nobel Prize. You know why? Because Nobel hadn't been born yet. All right. So the next big thing is, so we knew that chromosomes carried genetic information. And I don't know that I mentioned this, but we all have pairs of chromosomes. You get 2, okay? One from mom, one from dad. Well, what are chromosomes made of? Well, that's a whole big story in itself. And there were a couple of Nobel Prizes awarded for that. In 1910, chemists came up with, they found this stuff called nucleic acid, and they realized that it was made of these chemicals. And the 4 bases, adenine, thymine, guanine, and cytosine. Those are going to be the letters in the genetic alphabet. Genetic alphabet, it's simpler than English. There are only 4 letters, so you don't have a lot to memorize. And in 1962, the Nobel Prize was awarded to Watson and Crick. Probably you've heard of them. And the reason what they figured out was actually pretty clever, which was that they figured out how these bases were organized in chromosomes. And the key thing to note is that A always pairs with T. It's basically a double ladder setup. We call it a double helix because it turns. And the rungs of the ladder are the bases, and you've got the A always pairing with the T, and the G always pairs with the C. And what that means is if you cut the ladder in half, you can basically make the other half of the ladder with the information. And that immediately says, well, maybe that's how things are transmitted genetically. It gets cut in half, and then you rebuild the other side, and then you have two identical things from the one that you started with. It turns out, though, that DNA is only part of chromosomes, and chromosomes are actually built up on, if you look at the bottom, that's what the chromosomes that Hunt saw look like that. But it turns out that they're these DNA strands wrapped in proteins, and that was a couple Nobel Prizes for that, too. So anybody, young person here who wants to go into science, there's a lot of Nobel Prizes you can win. So this is, we're moving along here. Now, we know that, that chromosomes are made of DNA, and we have these factors that Mendel discovered called genes, but what exactly are genes and how does that work? So genes are segments of chromosomes that tell cells how to make a particular protein. Sometimes they tell cells how to make a particular structural RNA, but we're going to ignore that because for all of the diseases we're going to talk about, it's the protein that's the key. What are proteins? Proteins are molecular machines that make life possible. They're the little robots inside your cells that do the things that are needed to, that is life. It's very important to point that out. If anybody asks you, what's a protein? Proteins are molecular machines. The current estimate is that in humans, we have about 20,000 different proteins. Now, when I was a graduate student, we thought the number would be closer to 100,000 because we knew that in yeast, which is what I studied as a graduate student, there were 4,000. And nobody wanted to think we were only 5 times more complicated than a yeast cell. But it turns out we're not that much more complicated than a yeast cell, and we use pretty much all the same parts. So there are only 20,000 proteins in a human. Now, this is the take-home part of the talk. Everybody needs to know this. I hope most of you know it already, but I'm going to give it to you. This is called the central dogma of biology. And what happens is DNA has those long strands of bases, and that's the essentially plan to make all these 20,000 proteins. Think of it as the blueprint to make the building. And what has to happen is that DNA has to be read, which would be the same as, let's say an architect makes the plans then are Xeroxed, that's reading them, then that you get something called RNA. Looks just like DNA, but it's only got one strand instead of 2, and there's one substitute where the thymine is substituted for uracil, but basically it's the same. That's like the Xerox copy that then goes to the construction site where the protein is then, it says, this is how we make that machine. And that's what we call the central dogma. The key thing there is the fact they won the Nobel Prize was deciphering the genetic code. I said there's only 4 letters. How does that make protein? That was that one. That occurred in about 1960. So we're getting closer. This is the central dogma just shown a different way. We have our DNA. We have the RNA being transcribed. That means it's being copied from the DNA. It floats out of the nucleus of the cell, goes to this machine called the ribosome, and that then synthesizes a protein. Okay. Now let's talk about proteins for a minute. Proteins are chains of amino acids. We got 20 of them. There are 20 different amino acids that can make up proteins. And the way proteins work is the amino acids, at the one position will be a methionine, at the next position will be a valine, at the next position will be whatever the gene tells it to put at the next position. By the way, that's the proteins are made of amino acids. That's from one of the first Nobel Prizes in 1902. The important thing to note is that's all that the gene does, is it tells the ribosome what order to put the amino acids in. So you're like thinking, well, how does this chain of amino acids form this magical molecular machine that, he talked about earlier? And this is an example. This is actually the molecular structure of the cystathionine beta synthase protein, the one that you guys care about who have classical homocystinuria. And on the left there, I show you the amino acids, and on the right, that's what they fold up into, this beautiful structure. And for many years, we didn't really understand how an amino acid sequence folded up to make these beautiful proteins that can do dynamic interactions. But over time, and I listed 3 Nobel Prizes here, we've been able to figure it out pretty well now. So we now, for the proteins, for I think most of the homocystinurias now, we actually know the 3-dimensional structure, or we use AI to help us guess the 3-dimensional structure. And AI's gotten to the point where we can be about 92% accurate in our prediction. It was actually one of the first uses of AI, was this using it to solve the protein structure issue. So let's get now we're finally at HCU. So now we're moving on. So HCU is caused by the loss of key proteins that are involved in homocysteine metabolism. Classical HCU, as you've heard about, is mutations in the cystathionine beta synthase gene. The remethylation HCUs are caused by mutations in various genes involved in either folate or vitamin B12 metabolism. Okay? Both types of HCU are recessive disorders. You need 2 bad copies to have the disease. 1 is not sufficient. You need 2. You've seen this pathway before. In red here, I'm circling some of the key players. As you can see, homocysteine is kind of in the center there. We have, it gets converted back to methionine, and that uses this methionine synthase enzyme, so mutations in that cause a remethylation disorder. That enzyme requires this vitamin B12, and there's like 12 different enzymes that are involved in getting the vitamin B12 to methionine synthase. That's why that one's in red. MTHFR, I met somebody this morning who's has a grandchild with MTHFR deficiency. Actually, I met the mother too. That's over there. And then obviously CBS, that red line, that red dot should be over the CBS just below it. This just shows B12 metabolism. The only reason I throw this one up there is not to show you how smart I am, but to show you that it's a weird thing with B12. There are only 2 enzymes in the body that require B12 as a cofactor. The one that's involved in metabolizing homocysteine, and this other one, the methyl mutase in the mitochondria. But there's like 12 different genes that are involved in processing the B12 to get it to these 2 different places. And just as a scientist, I've always found that fascinating. Like, why are we expending so much energy? Are these 2 reactions that important? Apparently they are, or evolution wouldn't have done it this way, but that's how it works. Okay, now we're going to talk about how these genes cause disease. Most of the mutations in the homocystinurias are caused by what we call missense mutations. So here I've shown a picture of a fake piece of DNA. This piece of DNA has the DNA base is CAT, CAT, CAT. And if you go to that table I showed you earlier, that tells the cell, put a histidine in the protein. So then we have a bunch of histidines in the protein. What a missense mutation is, it's a single base that was changed out of the 3 billion base pairs that are copied. Sometimes mistakes happen. And if a mistake happens, and instead of an A, you get a C, well, that's going to change that amino acid from a leucine to a proline. And amazingly, that is enough to screw up that protein from folding up into that beautiful shape. It's that simple. One little change, it's like you're driving a car. If you have a bad wheel nut, that might be enough to have that wheel fall off and the car not work anymore. That's essentially what it is. So what I'm showing here is this idea that we have, we have the protein gets made, and then what we want it to do is we want it to fold into this beautiful folded protein, which is the one on the left. But sometimes if it has a missense mutation, it either folds incorrectly, like the one on the upper this, I guess, I don't know, upper left on mine. Or occasionally those misfolded, unfolded chains can form aggregates and they stick together. Almost always for these inborn errors of metabolism, it's the non-functional misfolded proteins that are the problem. Aggregates, though, are a problem in other types of diseases. So it turns out the cell has this network. We call it the protein quality control network. And so what happens is when the protein gets made, which is what's going on at the top, it then eventually folds. And if everything goes well, you get the thing on the left there. That's the folding you want. And these things in blue and yellow, they're called heat shock proteins, but they're actually chaperone proteins. They're proteins inside the cell to kind of help the protein get to the proper fold in a reasonable period of time. The proteins will eventually get there on their own, but these heat shock proteins, they're like, the nannies. They kind of help them do what they're supposed to do and get them there a little sooner. However, when the proteins are mutant, it oftentimes means that even in the presence of those heat shock proteins, they're unable to get to the right conformation, at least in any reasonable amount of time. And it turns out that your cells have a way to look at these proteins, and if they're not folded properly, they're like, you're a bad protein, you're going to the garbage disposal. And that's what they do. They go to this thing at the bottom there, which is called the proteasome, and that's literally what it is. It's the garbage disposal in the cell. These proteins that don't fold right, they get a little tag on them. The tag then tells the cell, send it to the proteasome. Proteasome chops it up, and the protein's gone. There was a Nobel Prize awarded to that. I'm very proud of that one because that was at my institute in 2004. Ernie Rose got that. And again, sometimes if proteins aggregate, then they don't go down the proteasome and they can go to this aggregation pathway. We're not going to talk about that. Almost always for the genetic diseases we're talking about, these missense mutations result in misfolded proteins. The proteins then get chopped up and aren't there anymore. They can't do anything, and you lack that function, therefore the enzyme doesn't work, the homocysteine builds up, and that's, as you heard this morning, that's the big problem with at least classical homocystinuria, or in the case, there are probably other problems with the remethylations, but I'm not going to go into that. So one of the things you can do is, or one of the things we showed you could do is, if you block that proteasome, so you can keep those proteins that haven't quite folded right from getting degraded, Eventually, they actually do make it. It kind of pushes everything back. Think of it like a pipe. If you got two places water can go and you block one of them, it pushes it to the other side. And that actually worked. So there's a drug, an anti-cancer drug called bortezomib. And if you look on the left in that I278T column where I show CBS activity or homocysteine, this was done in mice, you can see that in the mice that we give this drug to, we go from the UT, which are the untreated mice, have almost no activity. And you can see down there, they have really high homocysteine levels, around 350 micromolar. And you give the drug, and all of a sudden, a bunch of the mice have a lot of activity, and at least in some of the mice, their homocysteine levels go down. In red, it's a different proteasome inhibitor. It turns out different drugs work a little different, but the basic idea is the same. The problem with this, and so we were excited when we discovered this, but the problem was that it's an anti-cancer drug. When you block the proteasome, there are other side effects. It probably isn't something that a patient would, if you said, would you rather be on bortezomib or a methionine-restricted diet? You'd still rather be on the methionine-restricted diet. It's just not going to happen. We tried using it at low dose. It just didn't work. So you really have to give it at a dose that makes you feel bad. So the summary, though, from this part is that HCU are caused by mutations in key genes involved in methionine metabolism. Mutations cause loss of key enzymes in the pathways. Many mutations change only a single amino acid in the enzyme, and these prevent the enzyme from folding properly. And by manipulating the protein quality control network, it's possible to restore function of some of these mutant proteins. So now you are all, get your degree, you all have PhDs, you are ready now to understand everything else. But I felt like I had to be a little pedagogical there to help you guys. I want to make sure everybody's with me. So, first one we're going to talk about is molecular chaperone therapy. The idea here is we're going to use small molecules or drugs to stimulate proper folding of these mutant proteins that contain missense mutations. Remember, I just showed you that it is possible to take these mutant proteins and get them to work if you mess with this pathway. These small drugs, we like them because they can be given as a pill. So you'd have to take a pill every day. It doesn't seem like that big a deal. And to give you an idea, this is out there. FDA chaperone therapies have been approved for 3 different genetic diseases. You've got cystic fibrosis, Fabry's disease, and transthyretin amyloidosis. Don't ask me what that last one actually is. I'm going to give you an example, though, of the cystic fibrosis drug. It's in this beautiful picture. I said, Gemini, make me a picture showing how this drug works. And boom, it did. So basically, what this shows is that in cystic fibrosis, there's one particular mutation that makes up the vast majority of people with that disease. And in that, you're missing one amino acid. One amino acid, this phenylalanine at position 508, is missing. And what happens is that the protein doesn't fold properly, just like I told you. And in the case of cystic fibrosis, it gets stuck in an organelle called the endoplasmic reticulum. You have to go through the endoplasmic reticulum to get onto the cell surface, which is what this protein normally does. It's a channel that allows ions to pass through the cell surface. So they designed this molecule, lumifactor, that binds to this mutant protein in such a way that it helps it get to the right conformation. And by doing that, you now get these chloride ions flowing through in your lungs. It's been very successful. And this is the idea behind chaperone therapy. Now, for CVS, we don't have anything yet. One of my colleagues, Thomas Majtan, published this, I think last year or 2 years ago. he would look through all these FDA-approved drugs to see if there's a drug that maybe was used for something else that might be useful for CBS deficiency. And he found a drug called gavinostat that had an effect on the CBS enzyme. He's actually using the same mice that we use. And you can see that you can go from essentially no activity to a little activity. You're not back up to normal levels, but it might be enough to prevent some of the really bad things about the disease. The drug is FDA approved for Duchenne's muscular dystrophy. I don't know anything about people using it for CBS deficiency and whether there's some sort of trial. You'll have to talk to my MD colleagues about that. The caveats for chaperone therapy. The biggest one is it depends on the particular mutations in the individual. Not all mutations are rescuable, and they might not be rescuable by the same small molecule. So that's, you know, it's best when you have one mutation that causes the disease. In CBS, the most common mutation is, the two most common are G307S and I278T. Maybe for those it might be interesting. It's generally the rescue is only partial, and changing the chaperone environment inside the cell can cause problems for other proteins that are inside a cell, so there could be some side effects. So how do I want to say this? I'm not sure this is going to be the way to go, but I am kind of excited that there's a drug that's approved for something else that might be useful to look at. All right, enzyme replacement therapy. You're going to hear a lot more about that, I think, this afternoon. And it's pretty simple. We're going to replace the missing enzyme with a laboratory-produced molecule. And because proteins are broken down in the digestive tract, you have to give these as an injection, okay? They might be able to do it as a patch, but it's going to have to get inside your body somehow without going through your stomach. How long the protein stays in the blood determines how often injections are needed. It's been very successful. We have enzyme replacement successes have been used for Pompe's disease, Gaucher's disease, mucopolysaccharidosis, Fabry's disease, and most recently phenylketonuria. This is Gemini's version of how it works. I think it's reasonable. You take a piece of DNA called a plasmid, you put the human gene in it, you have the bacteria crank out a huge amount of this protein, you grow them in vats, that's the third one there, and then you break open those bacteria and you purify the protein and you're ready to go. It's that simple. It's not that simple, but you know, that's the base. It is that simple. It just, you got to make sure that there are no contaminants, and FDA is very particular, but that's pretty much how it works. And then you'll get the injection as a injection. I think in the case of the Travere drug, it's simply subcutaneous is fine. So that's similar to the type you get for GLP-1s for anybody that's on those. This picture that Gemini made thinks you have it in a doctor's office. I don't think so. I think you could get this, you could just do yourself at home. So it's very exciting, and I think we'll hear more about that this afternoon, and I'm looking forward to that. Now, this is pegtibatinase. This is my little slide. It's a recombinant modified CBS enzyme. It's very similar to the normal CBS enzyme. The changes have to do with making it a little bit more active and protecting it against your immune system. Early studies that were published said injection with 1.5 or 2.5 milligrams per kilograms 2 times a week could lower total homocysteine 57 to 67%. That seems like a pretty good number. And the drug was well tolerated. And as you heard, they're in phase 3 trials. I think they're recruiting, so they're looking for people. How well does it work? That's shown here. Um, the in the top line, the black line, that's the homocysteine levels in a group of 6 people untreated. So it starts out about 125 and ends close to that. And then in the 2 cohorts, and they're small cohorts, and only 3 people in each. So that's the only thing is this is a really small study. So you got to take everything with a little bit of a grain of salt, but you can see that they started out high and then they're lower at the end. And I think it's exciting. However, the caveat is it's going to be expensive. Enzyme replacement therapy for Gaucher's disease is estimated to cost $140,000 to $300,000 per patient per year. Who's going to pay for it? I think insurance pays for the Gaucher's one, so we're hoping they'll pay for the CBS one as well. Based on published data, therapy require injections 2 times a week. So, you have to be willing to inject yourself. I take Wegovy and I do that once a week. It's not a big deal. So if it's that easy, then it's fine. And the other question is, it's not clear if a 50% reduction in homocysteine is meaningful. So I think for the people in this room, what you really want is you want to know, well, if I take this drug, do I have to still be on the crummy diet? And I don't think the answer is clear because they, at least in the initial studies, all these patients are on their regular homocysteine diets. So I don't know how they're going to do that study.
They have an extension study. Once you complete the initial dosing, you can opt to stay in and modify diet and measuring.
Okay. So that's good. Thank you. But those are the things I would think about with this. All right. The next one we're going to talk about is gene therapy. So gene therapy, the goal is to introduce a good version of the gene to replace the mutant version. And currently this is done by genetic engineering a virus capable of infecting human cells. There are kind of 3 different flavors of gene therapy depending how the virus is introduced. So basically, this diagram kind of shows you the science behind it. What you do is you take a genome of a virus, and it's usually this adeno-associated virus. This is a naturally occurring human virus, doesn't seem to cause any problems. And what you do is you take its viral genome and you get rid of everything in it. And you replace that with the human gene that you want to express. In this case, in the case that I'll show you, it's cystathionine beta synthase. Then you get that fake genome packaged by giving what's called a helper plasmid. You don't need to know that. And you transfect some cells, you make some of this virus, and then you purify the virus, and then you can inject it and use it for various applications. There's 3 types of gene therapy, as I mentioned: ex vivo, local, and systemic. So the ex vivo example, there's a drug that's used to treat sickle cell anemia called Lyfgenia. And what they've done is they've modified the gene that's mutant in sickle cell anemia to prevent the cells from sickling. And the way they give this drug is they take blood from the patient, isolate blood cells, and then they infect them with the virus in a dish. And then they reintroduce that back into the patient. So we call that ex vivo because it's outside the body. And this is also the way that a lot of the current CAR T therapy works in cancer treatment. So for anybody who knows, if you have a severe type of cancer, this is now an option. And I should say a lot of this work has been done at the University of Pennsylvania right down the road here. In the local gene therapy example, it's instead of doing it outside the body, they're doing it outside the body, but they're injecting it into a particular organ. And the best example of this is this drug. I don't have the name of it on here. I forgot what it's called. It's for Leber congenital amaurosis, and it's the RPE65 gene. And they actually inject it in the eyeball. And this is a progressive form of blindness, and they literally can have patients that can now see because of this gene therapy. Lexterna, thank you. I should have been on here, and I somehow didn't put it on. And then the systemic form, and this would be the one that I think would be applicable to the things in this room. There's a drug that's just been approved, Zolgensma, for spinal muscular atrophy. And in this case, the gene for that is called the SMN1 gene, and the virus expresses that. And they give it as a single IV dose for children under 2 years of age. And I'm just going to read you, this is from the studies that were done. At age 14 months, 91%, that was in the US study, and 97% in the European study, of treated patients were alive and free from permanent ventilation, as compared with 26% in the historical cohort. I mean, that is spectacularly good. And that's what we want to see, I want to see, people with gene therapy, you guys can go do anything you want, and you're no different than anybody else, and you can eat as much steak as you want. We have done gene therapy with our mice, and these were studies we did in collaboration with our collaborator at Cornell, but they were done here at Fox Chase. And you can see here that in blue, that's the serum total homocysteine on that side. That on the untreated mice. And then in red, you can see the total homocysteine on the treated mice. In black is the total homocysteine on the control mice. So it's not quite as good as the control, but it's pretty good. And I should mention, we did these studies out for a year. A mouse only lives about 3 years in the lab. So this would be one dose to get you to age 30 would be pretty good. Maybe we can give you a second dose when you're 30, but it's still even really good at 1 year. And the mice put on weight, they look better. You can see, look at the bottom there. So the one in the middle is the control mice. That's what our homocystinuria mice look like if they're not on special diet. And they're on the same non-special diet, but with gene therapy, they're totally cured. So it's very exciting. So the pros of gene therapies are one and done. It seems to be highly effective. By far and away the best treatment that we've looked at in my lab, and we've looked at a lot. Cons are immunogenicity could be a problem. Like you could develop immune reaction to the virus, which might prevent you from getting a second dose later if you needed one. The big, I think, drawback in the field now is people are really concerned about what we call unwanted integration events. Normally, these AAV genomes don't integrate into your chromosomes, but occasionally they do, and people are worried that could cause cancer. I think that worry is overblown, but it's been a big problem, I think, in the field. And then lastly, cost is a big issue here. This could be, for these other gene therapies, they range in price from $850,000 to $3.5 million per patient. That being said, insurance will pay for it. So, in some cases where it's been looked at. So how that's going to work with CBS, I don't know. Now, the last one is kind of, yes, we're almost done. The last one is kind of the most futuristic of the things, and it's involving this. You've probably read about it in the news, CRISPR. A Nobel Prize was recently awarded for that in 2020. And basically what CRISPR does is it allows us as genetic researchers to make very precise changes in the DNA of living organisms. In fact, I'm using this technology right now to make a mouse to study cancer in my lab. And I don't want to go too much into the detail. It was discovered because it was discovered as a bacterial system, and they've been able to modify it. So now they can use it on any type of cell. And what happens is that you can, by combining CRISPR, which allows you to make a very precise cut at a specific point in the DNA, and then you add a little piece of DNA to use to repair that cut, you can make very targeted modifications. So in theory, let's say you had a G307S mutation for classical homocystinuria, you could, in theory, just fix that one mutation. This is just showing we used it to make mice, so you can just inject it in mice. I don't think we need to go into that. It's exciting because the world's first patient was treated with CRISPR gene editing last year at CHOP, and it showcases this. And what they did is, this is another inborn error of metabolism caused by mutations in the CPS1 gene. They get really high ammonia levels in the blood as a result of this mutation. And they identified a missense mutation. Actually, it's a nonsense mutation. I don't know why they said missense. In the CPS1 gene that in this one patient. And so they made a particular CRISPR molecule to create a nick in the DNA that, when repaired, would fix the mutation. They took this CRISPR molecule and enclosed it in a lipid nanoparticle. So they're not using a virus here. They're actually using just like an artificial lipid. And then they injected it, and they were able to get it to go to the liver of the patient, integrate, and they corrected enough cells in the patient that their plasma ammonia levels went down significantly. And so what you can see on the chart here is probably this urinary oxalate is probably a better thing. So you want that to be high. And in the baseline, it's very, very low. And then when they gave it in 2 doses, and you can see now most of the readings are much, much higher. And conversely, the ammonia goes down. And so it's really exciting. I mean, this is pretty much state of the art. Is anybody doing this for any of the remethylation defects right now? No. But there's no reason this wouldn't work for any of the diseases in this room. So in this part, I just want to sum it up. Chaperone therapy, ERT, gene therapy, and gene editing are all potential therapies for HCU. For classical HCU, enzyme replacement is clearly the furthest along. Preclinical testing in mice suggests that gene therapy is the most effective. The rate limiting factor moving these therapies from the lab to the clinic, frankly, is money. That's I hate to say it, but that's how it is. And I'll just stop here, and I want to thank Margie for her support for all these years. This was my group from a few years ago that did a lot of the work. Don't forget to thank the mice. They've given a lot of their lives for people in this room. And HCU Network America and the NIH also NIH gave us the bulk of the money to do the work. So thank you. And I'm happy to answer any questions.
Just curious, you mentioned the enzyme replacement therapy that's approved for PKU patients. Do you have any sense of what percentage of this PKU?
I didn't do a deep dive to look at that, but I'm sure somebody here might know. Because that's probably the most Yeah, exactly. It's your closest ally. Exactly correct.
It's complicated, so they're slowly introducing it into the patient. I think it'll grow much higher.
And when did they receive FDA approval? Any idea on that?
I think it was maybe 18 months ago.
Still relatively
18 to 24.
Was there a question in the back? Yep.
Yeah, on the cost of the gene therapy, like $2 million, $3 million. There was a New York Times article a couple months ago about how like rare diseases are so rarely researched by pharmaceutical companies for so few patients. But do you think like doing one big surgery like that that's really helpful but expensive is something that like companies look at more?
So I think that's why this organization that Margie's put together is really important. I think that they'll do it. They'll do it if they can make money at it. And in order to, they need to know how many people are out there. How big is this patient population to make it worth their while? One of the reasons gene therapy is so expensive is the actual cost to produce the amount of virus is very expensive. You know, it's not cheap. But the other reason it's really expensive is, you know, nobody would, the estimate that I've heard from my colleague at Cornell, who knows a lot about this, is that to take the gene therapy that we have our data for from our lab to get it to the point where we can get through what's called phase 1 clinical trial, a minimum of, people say he's lowballing, but $10 to $20 million. And that's the type of money that is going to be required just to get it through phase 1. You know, maybe a 1-2, you know, but then you still got to do the phase 3 study to get the final approval. So frankly, I think not only HCU America, you've got to scour all of these, you know, I don't know if there's an HCU Qatar, but I would go there next. And HCU Australia exists. The network in Europe is good. Who knows how many homocystinuria are in China or India? But the more, you know, if you can show them that there could be over 1,000 patients that could get this, you might be able to convince somebody that it's interesting. I don't think there's any scientific reasons why this won't be successful for classical HCU. For the remethylation disorders, I think there's some more unknowns. We're not really, for classical HCU, it seems like if you can lower the homocysteine, that's all you need to do and you'll be good. But for the remethylation defects, it seems like that the low methionine could be more the problem, and that might require something that actually gets into the nervous cells to restore their balance of, I didn't talk about it all, but acetylmethionine and acetylhomocysteine, this methylation pathway is probably what the problem is there. And that'll be trickier, but I don't think it's impossible. I know Dr. Vendetti has some gene therapy for one of the cobalamin disorders that looked, or actually it was the mutase disorder, but it's a related disorder and it looked very promising. So I think that could work as well. So I don't want to discourage that. It'd be good though to have somebody try gene therapy on MTHFR deficiency. I totally agree. Yeah. You know, that's an interesting question. I don't know how they did that. They got a one-off there. Right. They got like a compassionate use probably. And this is part of the problem is you guys aren't sick enough. I mean, in terms of if you're about to die, like in cancer, to get a cancer therapeutic, you can get compassionate use sometimes for a new use of a drug or something. But it's going to be hard here. You're going to have to do a clinical trial. I appreciate Thank you for your attention.